Welcome to the debate. When we evaluate the success of a major city, we often start by looking at the sheer volume and, I guess, the variety of its population.
Right. Imagine standing on a skyscraper, right? You're looking down at the bustling streets. You see millions of people moving about. You've got different professions, different cultures, different daily routines.
It's a massive, tangled web of activity.
Exactly. And intuitively, we equate that incredible density, that staggering variety of life, with urban vitality and resilience.
Yeah, well, it feels completely instinctive, doesn't it? More people, more diverse professions, more overlapping economic activity. You just assume that if one industry falters, another is right there to keep the city running.
It feels like a thriving, unstoppable ecosystem. But then you take the elevator down to the street level, and you realize that simply counting the population tells you absolutely nothing about the actual infrastructure.
Well,
It tells you nothing about the crime rate or, whether those people are actively working together symbiotically or if they are just fiercely competing for a dwindling supply of resources.
I see your point.
Right, because the sheer size of the crowd is just a number. It completely obscures the actual mechanics of how the city survives.
So today, we are turning our attention to the microscopic metropolis within and around us, the microbiome. Specifically, we are examining the role of microbial diversity in ecosystem and human health.
A massive topic.
Definitive marker and fundamental driver of a healthy microbiome? Or is diversity an oversimplified metric that obscures the more critical factors of specific microbial composition, function, and inter-kingdom interactions?
I think we both know where we stand here.
Oh, definitely. I will be arguing that a highly diverse microbiome is the essential engine of health. It provides functional redundancy, resilience to environmental changes, and rigorous resistance against pathogen invasion.
And I will be arguing that relying on broad diversity is mathematically elegant, sure, but biologically flawed. When we actually look at how these microscopic ecosystems function, specific microbial identities, precise ratios, and functional interactions across different biological kingdoms matter far more than a raw diversity score.
Let's lay out our core frameworks to start. From my perspective, biodiversity is the absolute gold standard for ecosystem health. And this applies whether you are looking at the Amazon rainforest or the human gut.
So you're equating a rainforest to a colon.
I mean, ecologically speaking, yes. High diversity means that a vast array of metabolic functions are being performed simultaneously. This generates a biological safety net. We call it functional redundancy.
Meaning if one species goes down, the ecosystem doesn't just instantly collapse.
Exactly. Imagine your diet suddenly shifts. Say you travel abroad and drastically change what you're eating.
Like eating entirely unfamiliar carbohydrates.
Right. If one specific bacterial species that helps digest those complex carbohydrates gets wiped out by that shift, it is not a catastrophic event in a diverse gut. Why? Because another distinct species is already there ready to take over that exact metabolic job. Furthermore, a highly diverse, fully occupied community heavily resists invasion. When all the biological real estate and all the nutrients are actively being utilized by a rich tapestry of native microbes, it becomes incredibly difficult for an exotic pathogen to find enough resources to establish a foothold.
I'm sorry, but I just don't buy that. Let me tell you why. The concept of diversity, particularly how we quantify it in actual research, is largely a mathematical approximation.
An approximation?
Yes, tools like the widely used Shannon Diversity Index are just equations. They account for richness, which is the raw number of species present, and evenness, which is how well distributed those species are.
Which are critical metrics.
But treating microbes as equal numbers in an index completely ignores the profound reality of strain-specific pathogenicity.
You're saying a number doesn't tell you if the bacteria are actually friendly.
Precisely. A high diversity score does not inherently equal a healthy gut. For instance, you can have a biological sample that generates a beautifully high diversity score, yet still features a dangerously imbalanced firmicutes to bacteroidetes ratio.
Ah, yes. Let's unpack that ratio for a moment.
Sure. Firmicutes and bacteroidetes are two massive groups, or phyla, of bacteria in the gut. Firmicutes are incredibly efficient at extracting energy, literally calories, from the food we eat.
Right, whereas bacteroidetes are generally associated with a leaner phenotype.
Exactly. So if your ratio is heavily skewed toward firmicutes, your gut is hoarding calories and promoting systemic inflammation. Mathematically, the ecosystem might look as diverse as your rainforest analogy, but biologically, that specific ratio indicates a dysbiotic gut that is actively driving metabolic syndrome and obesity. We absolutely have to look past the raw numbers to the specific identities of the microbes.
I see why you think that, but let me give you a different perspective. Diversity is not just a passive state or a simple head count. It is the active driver of evolutionary success through cross-feeding and mutualism.
I mean, I agree mutualism exists, but
But the sheer variety of species is what creates the chemical complexity necessary for those specific identities to even survive in the first place. Let's drill down into how we actually measure this, because, how we measure diversity dictates our understanding of it.
absolutely.
Alpha diversity, the richness and evenness you mentioned, is a crucial baseline. But to appreciate it, we have to measure it correctly, using high-throughput 16S RNA gene sequencing.
Which is essentially like scanning the barcodes of the bacteria to see who is present in the sample.
Right. But historically, we weren't scanning deeply enough. Imagine you have a massive burlap sack filled with 1,000 pieces of candy, right? In 6 different colors.
Okay, I am picturing the candy.
If you only reach in and pull out 5 candies, you might conclude there are only 3 colors in the whole bag. You severely underestimate the reality of the bag.
Because your sample size is too small.
Exactly. In microbiology, if you don't sequence deeply enough, you miss the rare underlying species. When we utilize deep sequencing to find the true plateau of a community's diversity, it proves that a diverse gut isn't just a handful of dominant strains. It is a wildly complex, interacting network.
I come at it from a completely different way. I mean, I completely agree that sampling depth matters. We absolutely must use statistical techniques like rarefaction.
Which is downsampling for our listeners.
Right. Rarefaction essentially means downsampling our data so we are comparing apples to apples, ensuring a study that sequenced a million barcodes can be fairly compared to a study that only sequenced 10,000.
Agreed.
But my contention is that simply counting the colors of the candy entirely ignores what those colors actually do inside the human body.
But doesn't the sheer variety inherently suggest a more robust, stable structure?
Not necessarily. Let's look at the Shannon index again. The math multiplies the proportion of a microbe by the natural logarithm of that proportion. You typically end up with a score somewhere between 1.5 and 3.5.
Sure, standard ranges.
That score tells you exactly how mathematically diverse a gut is, but it tells you absolutely nothing about how healthy it is. Consider the oral microbiome in the context of oral squamous cell carcinoma, mouth cancer.
Okay, what does the diversity look like there? I imagine it collapses.
That's where we find a fascinating paradox. Studies profiling the oral microbiomes of these cancer patients versus perfectly healthy controls reveal that the cancer patients actually exhibit higher alpha diversity.
Wait, really? So the disease state is more diverse?
Yes, they have a richer microbiome in the localized tumor environment.
That is wild.
But when you move past the diversity score and look at the actual composition, you see a massive abundance of specific bacteria, like Fusobacterium nucleatum,
Ah, yes, a notorious bad actor.
a highly known driver of carcinogenesis. It literally binds to host cells and creates an inflammatory microenvironment that promotes tumor growth. In this specific context, an increased richness score is actually indicating the aggressive proliferation of pathogens.
Okay, I see.
It is a highly diverse, incredibly dangerous tumor environment. If we are only looking at the diversity index, we are completely blind to the pathology.
Look, I will acknowledge that specific aggressive pathogens can distort the mathematical picture in localized advanced disease states like cancer. But let's zoom out. Let's look at the broader mechanics of how microbial ecosystems assemble and sustain themselves over time. This brings us to the diversity begets diversity model, the DBD model.
The idea that diversity naturally breeds more diversity?
Exactly. If we look at the Earth Microbial Project, which, by the way, is a massive, standardized global sampling effort utilizing 5,000 sequences from 2,000 carefully rarefied samples, we see a clear, positive feedback loop.
A massive data set, undeniably.
And it shows that initial diversity actively creates brand new ecological niches for other microbes to assemble into.
Right, the data shows a positive slope between the diversity of a core lineage and the diversity of the background community.
Precisely. And what is truly fascinating is the mechanism behind it. Why does this happen? The data shows that bacteria with larger genomes, we are talking up to 14 megabase pairs, which is a massive genetic construction manual for a microbe, experience significantly higher DBD slopes.
Because they can do more things?
Because those larger genomes encode a much wider array of metabolic functions. They are secreting a massive variety of chemical byproducts.
So one microbe's waste literally becomes another's food?
Exactly. It's called cross-feeding. By metabolizing nutrients and excreting unique byproducts, these larger microbes are literally constructing new niches for entirely new species to migrate into.
That's an interesting point, though. I would frame it differently. It's not a zero-sum game. The microbes build the city they live in. They expand the available real estate through their diverse metabolic output. The Earth Microbiome Project data is remarkable. I don't dispute that at all. But you are omitting the second half of that exact same ecological analysis.
Which part?
The data also clearly supports the ecological control model.
How so?
Well, yes, diversity begets diversity initially, but the data shows that this compounding effect actually plateaus in highly diverse free-living environments, like soil or in a mature adult gut.
Well, obviously it can't grow to infinity.
Exactly. It doesn't go on infinitely. Once those newly created metabolic niches are filled, strict ecological control takes over. The environment simply cannot support endless expansion. But more importantly, in this entire city-building analysis, you are still only talking about bacteria.
Well, bacteria do make up the vast majority of the biomass and the metabolic activity in the microbiome.
They do, but they do not act alone. Assuming they do leads to fundamentally flawed conclusions about health. Let me bring in Dr. Marie-Claire Arrieta's gnatobiotic mouse models.
Gnatobiotic meaning these are mice born in perfectly sterile bubbles with absolutely no natural microbiome.
Exactly, germ-free mice. The researchers deliberately colonized these mice with a defined low-diversity bacterial consortium called oligo-MM12, so they knew exactly which bacteria were in there.
Down to the exact strain.
Right. Then they introduced yeast fungi into the gut. Now, based on the diversity begets diversity model, if diversity is always additive and always beneficial, you would expect the bacterial richness to at least stay the same, if not grow, from new cross-feeding opportunities.
And what actually happened?
The introduction of the yeasts actually reduced the bacterial richness by week 4. The bacterial diversity went down.
Because the fungi essentially outcompeted the bacteria for the limited resources in the gut?
It is much more profound and simple competition for food. Despite lowering the overall mathematical bacterial diversity, the co-colonization of yeast and bacteria created an incredibly powerful synergistic immune response in the host mouse.
How did they measure that immune response?
They looked at the spleen. The mice developed differential proportions of critical immune cells, specifically macrophages, which act like the immune system's garbage collectors,
Right.
and FOXP3 regulatory T cells, which act as the peacekeepers that prevent autoimmune overreactions. The host's immune system was trained far more effectively by the crosstalk between the two biological kingdoms than it ever was by maximizing bacterial diversity alone.
That's fascinating. So you're saying the presence of the fungi fundamentally changed the behavior of the bacteria?
Radically. When researchers looked at the metaproteome, which, isn't just looking at the DNA barcodes, but looking at the actual proteins the bacteria are actively manufacturing, they found that the bacteria had fundamentally changed their protein expression just because the fungi were in the neighborhood.
Wow.
True ecosystem function and host immune development comes from this inter-kingdom crosstalk. It doesn't come from hoarding the highest possible number of bacterial species.
I hear you, but I'm not convinced by that line of reasoning. Because while inter-kingdom crosstalk is undeniably an important piece of the puzzle, A foundational loss of broad bacterial diversity remains a remarkably consistent, highly reproducible hallmark of systemic human disease.
But is it the loss of generic diversity or the loss of specific functional microbes? That's the million-dollar question.
Let's look at the mechanisms in human clinical applications. In a massive study of 871 female twins, researchers found that a lower alpha diversity in the gut is significantly and independently associated with hypertension, or high blood pressure.
Let's break down the mechanics there. How does a less diverse gut raise your blood pressure?
The study identified the exact cascade. When the overall alpha diversity drops, the ecosystem loses its structural integrity. Beneficial genera, like Rumina Clostridium 6, precipitously drop.
Okay.
Now, Rumina Clostridium 6 is functionally mapped to 83 different metabolic pathways, and that includes those that generate short-chain fatty acids.
Ah. And short-chain fatty acids are crucial signaling molecules.
Right, they actually enter the bloodstream and help blood vessels dilate, naturally regulating and lowering blood pressure. So when you lose the diversity, you lose the short-chain fatty acids and blood pressure spikes.
And I assume other microbes fill that void?
Exactly. Simultaneously, opportunistic microbes like Erysipelotracheae rise and dominate the space. The overall collapse of the diverse community architecture is what allows these specific dysbiotic shifts to occur in the first place.
So you're arguing the diversity itself is the shield that keeps the ecosystem stable.
Exactly. We even see this macroecologically. Experimental warming and extreme global temperature fluctuations significantly deplete the phylogenetic diversity of microbiomes across terrestrial and marine hosts.
Global warming effects, yeah.
And when that broad diversity is depleted by a shock to the system, the host organism's physiological performance is directly threatened. Why? Because the functional redundancy required to survive environmental stress is simply gone.
That's a compelling argument, but have you considered that our clinical attempts to artificially boost or maintain that broad diversity have largely failed?
I assume you're talking about probiotics.
Yes, probiotics. If restoring broad diversity were the ultimate clinical cure, probiotics would be our magic bullet. We could just swallow a pill packed with 20 different bacterial strains, and boom, we rebuild the rainforest.
Which we know isn't quite how it works.
Not at all. A rigorous systematic review analyzed over 11,000 screened articles looking at administering probiotics during antibiotic treatment. The explicit goal of the treatments was exactly what you are advocating for, to prevent antibiotic-induced low-diversity dysbiosis.
And the results were negligible?
They were a complete failure. Quantitative data synthesis for the Shannon Index, the Chow-1 Index, which for the audience estimates total richness by factoring in rare species, and observed taxonomic units revealed absolutely zero significant difference between the probiotic groups and the control groups.
Why do you think that is? I mean, if you introduce billions of diverse microbes, why doesn't the mathematical diversity increase?
Because they are like tourists arriving in a city where all the apartments are already rented out. Or, more accurately, they simply lack the highly specific metabolic partners they need to survive.
The mutualism aspect.
Exactly. We are ignoring the underlying evolutionary biology of how microbes actually live. Consider the Black Queen hypothesis.
Named after the card game Hearts, right? Where players actively try to avoid taking the Queen of Spades because it costs them heavily in points?
Exactly. It is a brilliant, totally counterintuitive concept in microbial ecology. The Black Queen hypothesis posits that certain bacteria actively shed genes over evolutionary time. They intentionally delete parts of their own DNA.
Which on the surface sounds like a terrible survival strategy. Why would a microbe throw away its own genetic toolkit?
Because replicating DNA takes a massive amount of energy. If you are a bacterium living in a densely packed gut, and the microbe right next door to you is constantly leaking a vital amino acid, why should you waste your own energy manufacturing that amino acid?
So you just delete the gene for it and steal from your neighbor. Precisely. You streamline your genome, you become incredibly small, incredibly fast at reproducing, and highly efficient. But there has to be a catch. There is a massive catch. By losing those genes, you become permanently dependent on that specific neighbor to produce the metabolites you can no longer make. So your survival is literally tied to theirs. Completely.
It is a highly specific, lock-and-key, gene-sharing network. We call it precision dependency. If a harsh course of antibiotics wipes out the specific partner of a streamlined microbe, that microbe dies too.
Even if it wasn't directly targeted by the antibiotic?
Exactly. And no amount of generic, store-bought diversity in a probiotic pill will save it, because the generic probiotic doesn't have the exact metabolic key to keep the streamlined microbe alive.
That makes sense.
Moving past basic taxonomy and the outdated concept of simply counting species, and instead looking at this incredibly intricate strain-level variation and specific metabolite exchange, is the only viable path forward for precision medicine.
That is a fascinating mechanism, but let's summarize where this debate leaves us today.
Sure.
My position remains that while specific microbial identity, like knowing exactly who holds the metabolic key, is certainly important, a foundational baseline of high diversity is non-negotiable. It is the prerequisite for health.
Right.
Without a high degree of richness and evenness, the ecosystem simply lacks the structural integrity, the functional redundancy, and the cross-feeding architecture required to survive environmental shocks. Whether that shock is a harsh course of antibiotics, a dietary overhaul, or an invading pathogen, diversity is the engine that drives ecological stability. And my conclusion is that diversity as a standalone concept or a mathematical score is a remarkably blunt instrument.
Fair enough.
It gives us an illusion of understanding. An ecosystem's health and its actual physiological impact on the human host is dictated by the precise ratios of its inhabitants, like the crucial balance of firmicutes to bacteroides.
The metabolic hoarders versus the lean phenotype.
Right. It is governed by strain-specific metabolic functions, precision dependencies like the Black Queen hypothesis, and crucial inter-kingdom interactions with fungi and viruses. A simple diversity score completely fails to capture the actual biological reality of these complex, microscopic economies.
Yet I think we have reached a vital point of convergence today.
I think so too. Whether we view diversity as the foundational driver of health, or merely as a superficial metric that hints at deeper interactions, we both firmly agree that the future of microbiome science must aggressively move beyond simplistic bacterial headcounts.
Absolutely. The field is maturing rapidly. We must transition into multi-omics.
Meaning looking at the whole picture.
Right. Metagenomics to see the DNA blueprints, metaproteomics to see the actual proteins being built, and metabolomics to measure the chemical byproducts being shared. We have to understand not just who is there, but what they are actually doing.
And we must expand our focus to fully integrate the incredibly complex relationships between bacteria, fungi, and viruses.
Exactly.
We began our conversation today by visualizing a bustling city, debating whether the sheer volume of its inhabitants was the true measure of its success. As we've explored, the true vitality of the microscopic metropolis within us relies just as much on the invisible, deeply intricate networks of cooperation, genetic dependency, and ecological control. We will leave it to you, the listener, to ponder the true definition of a healthy microbial landscape. Thank you for joining us on The Debate.